Hello, my fellow Krembil avatars. I'm Mary Pat McAndrews, and I'm the chair of the Krembil Trainees Affairs Committee, and I'd like to welcome you all to our second and hopefully final pandemic Krembil Research Day. As in the past 20 years, this is a day to celebrate the wonderful research of our trainees. And to kick off the proceedings, I'm happy to introduce Dr. Brad Waters, the Executive VP of Science and Research at UHN, for formal opening remarks for us. So, Dr. Waters, over to you.
Well, thanks, Mary Pat, and it gives me great pleasure to welcome everyone today to the Krembil Research Day. This is a terrific event at UHN, and I think Krembil does a really outstanding job at highlighting the research of all of our trainees as a means towards furthering and fostering further communication and interactions. And this has been a really successful day over many years. And as Mary Pat said, for the second time, we're doing it virtually. The event is something that we look forward to at UHN because it's an opportunity for us to connect with each other and a chance to speak with each other, to think about new ideas and to think about, how our individual projects can benefit from input from each other. And this is really what the value of a research institute is all about. It's about what we bring to support and impact each other's work and what each other can bring to help our own. And it's an extremely important part of science. And if there's one thing that we've learned over the past year in this pandemic, it is that connection piece, that collaboration is really essential for our progress. And with our COVID restrictions, I know that many of you and all of us really have had to pivot and move to a new working model and to try and stay productive and, and to be agile and flexible and resilient. And I think, I've been extremely impressed by the ability of our scientific community to do that, but it has been challenging. And when we look back over the past year at our progress, many of the metrics actually look outstanding. We've brought in lots of new grants. We've published more papers this year than ever in our history, and fantastic high impact publications. And maybe some of that is, everyone going home and cleaning out the closet a little bit and writing up those papers when they've had a chance to be at home. But we also recognize that there's something that we've been missing, and that interaction, that energy, that stimulus that we all give each other is part of that, and it supports the work that we're going to do tomorrow and next year and in the future. And so we're worried about that, and we want to make efforts to bring people back together, to generate that excitement, to generate that collaborative spirit, and we're going to be working hard on that as we move towards a more return to normal. But today is a day to do that too. And that's really the purpose of Research Day. We're doing it in the context of a virtual platform this year, but I encourage everyone to kind of think about that. And it's a really important part around our efforts to stay productive, to create impact, and to continue to reach new highs in what we can achieve together. And despite the challenges that we've all faced and that you've all faced this year, professionally, personally, financially, there are many of these, and many of them are hidden from each other that we've all faced over the past year. What we have accomplished, I think, in that context is nothing short of incredible. First of all, I want to say how thankful I am to all of the efforts that everyone has made to keep each other safe. And, we went through a shutdown in research, we went through a restart, we put in lots of tough conditions that I know have impacted the way we can work and the way that you all can work. But I'm extremely proud of the fact that we have not had one single instance of an infection of a staff member in research that has occurred at work. We've had cases where research staff have gotten COVID and been infected in the community, but we have no known cases of infection at work. And it's a reflection of how serious everyone has taken the public health measures and the other measures we put in place to keep our workplace safe. And I think we have, amongst the safest work environments anywhere in the country or anywhere in the world for that matter. So that's terrific, and we all deserve a piece of the credit for that. But on the research front, we've also made some really impactful and impressive strides forward. I talked about everyone's current work. We've also, contributed to the global effort to address COVID-19. And, that includes efforts in infectious disease and diagnostics, but it also includes efforts in neuroscience and cardiovascular disease and in, all mental health and all various aspects of how COVID-19 is affecting society. And just to give you, a bit of a metrics around that, UHN has launched more than 250 COVID-related projects that have received REB approval. So these are clinical research studies of some kind. We've launched 34 clinical trials, and we've brought in $38 million in new funding, unanticipated funding, specifically for COVID-related research. Over 7 million of that has come from our foundations, and nearly 30 million from external peer-reviewed organizations. So an incredible level of new activity. We've also were successful in bringing in over $40 million from the federal government as part of our advocacy efforts last year around asking the federal government to support research at research hospitals because of the impact COVID was having on our finances. So UHN led that effort. And we had lots of people that contributed to that, and it brought into UHN over $40 million last year, which helped us finish last year in a balanced position with zero impact of COVID on jobs or on positions, and something that at this time last year we were still struggling with. We've also published more research than ever before, as I mentioned, and at the Krembil, we've seen cutting edge, high impact papers on lots of areas, on repeated concussions, neurodegeneration, immune cells that play a critical role in spinal arthritis, new pathways that govern brain development. We established the Max Planck University of Toronto Center this year for neuroscience and technology, an exciting area to unite research in engineering and physics and neuroscience and neuroinformatics and in medicine, and bringing our strength in the translational aspects of that to some of the more fundamental biology with our partners. We also founded the Schroeder Arthritis Institute this year with a historic gift from the Schroeder family to help set the stage for the future of that institute and position it on the world stage as a leader in arthritis research. And many of our world-class researchers were also recognized internationally with prestigious awards. Another highlight of the year was the Gairdner Symposium on Alzheimer's disease. This is a very prestigious symposium offered by the Gairdner Society. This was a terrific success, and it was strongly impacted by the work of the Krembil office and was dotted with lots of input and discussion from research at the Krembil Brain Institute itself. We also launched the 2020 Vision magazine and so much more. So when you listen to today's presentations and you kind of walk through this virtual space and bump into each other's avatars, take a moment to remind yourselves of this community that's been created here at Krembil. That sense of community is a cornerstone of how we want to operate here at UHN as a leading research hospital. And remember, as we've tagged our strategic plan, we're in this together. Our success is a collective success. And this is a chance today to feel part of that wide variety and broad research impact that we're creating. And finally, I just want to thank the Krembil's Trainee Affairs Committee for once again putting a terrific lineup together today, and also the Krembil admin team who do an incredible job at this. It's the envy of UHN, and all of the logistics that go behind that. We obviously may not have expected a second virtual research day, but it doesn't diminish the excitement. And we're all watching the impressive results on vaccination, the impressive results on reduction in cases, and actively planning our return back to normal. So with that, I want to again, just encourage you to enjoy today. And I'm going to introduce a short video now to hear from some of the others in our community who would like to also say a word of thanks to all of you. Thank you.
It was a long time ago, but believe it or not, I can still remember when I was a trainee, and we know who does all the work. I really do appreciate trainees, and I know how valuable you are to the research mandate of Krembil.
I want to thank all of our trainees for all of their hard work, commitment, and enthusiasm for the research that we do.
Trainees have often said that I inspire them, but the fact is, they inspire me. They are the lifeblood of research.
I moved to the Krembil Research Institute about a year ago, and I know firsthand how important trainees are.
To all the Krembil trainees, a big, heartfelt thank you.
Our discussions and sometimes heated interactions have been fun and enlightening, pushing me in directions I may not have gone otherwise. You know what I mean.
I love mentoring trainees because they ask those how about and why not questions.
I always look forward to hearing new ideas, the vibrant discussions during lab meetings, and helping me to mentor trainees as they navigate challenges.
My greatest joy is in nurturing the development of my trainees and to see how passionate they become in their own pursuits of new knowledge, each of whom then follow their own unique path towards fulfilling careers.
My goal really is to create an environment for really smart people like the trainees to sort of create the next generation of science.
I was very fortunate to have mentors, supervisors that had one thing in common. They created a very friendly, open environment without overbearing pressure. I think if you can create that perfect environment, the trainees will flourish and bring your research to life.
I really love mentoring trainees. I've learned so much from them about new statistics and new music. It's a wonderful feeling to have a piece of yourself go with them on the rest of their journey.
But during COVID, I feel like all of them have really stepped up.
You've all shown your dedication by still wanting to come in and do experiments.
Dealt with the restraints of shift work, dealt with reagent shortages, and still produced and delivered data.
I'm so proud of each and every one of you for your stellar achievements over this past year. You have ensured that science progresses during these tough times.
Trainees have shown amazing commitment, strength, and dedication during this pandemic, making sure the research project were pursued further.
Because without you, we wouldn't keep making discoveries.
And the work you're doing will improve the quality of life for an incalculable number of people in the future. So whenever you're feeling down, please remember that you're making a huge difference in this world and that you are a part of the Krembil family. We're all working toward the same goal, to improve life.
We're a community made especially obvious during these COVID times. We're all in this together.
I really want to thank them, and I owe really everything to my trainees.
You guys are doing amazing work. Keep up everything that you're doing. You really make a difference. Thank you so much.
I'm really proud of you. I mean it. Thank you.
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 Thank you. Merci.
Thank you.
Thank you so very, very much.
Thank you for all that you do. We couldn't do it without you.
Once again, thank you very much for all your grace and grit that you showed, which is always so important in research, and no more so than over the past 15 months. So from all of us, as was said. And so I just want to give you a little bit of additional information. As you've read in our program booklet, we're using a mix of Zoom for the oral presentations and GatherTown for posters. I hope you'll find the latter a fun and interactive experience, and you'll see that you can travel to a poster and directly talk with the presenter. You'll also note that you can chat with others based on your proximity in the hallways or in private chat rooms that you enter. Any problems with the platforms, no worries, because the Krembil admin team has our backs, and they're available through direct messaging throughout the meeting. So hopefully we'll all have a wonderful and engaging day. And to start with our first block of presentations, I want to invite our chair, Dr. Jen Wither, to the podium.
Hi, welcome to the morning oral presentations. And what I would like to say is that each presentation is going to be 10 minutes, and then we'll have 5 minutes for questions. If you have a question, I would like you to enter it in the chat function. If you wish to ask your question orally, I would just say, enter that you want to ask your question orally next on the chat function, and then I will invite you to unmute yourself. So our first presentation this morning is from Scott Rich, who works with Tawfiq Valiante, and I invite Scott now to start his presentation.
Awesome. Thank you so much, Dr. Wither. I'm very excited to present new work from our group about how lost neural heterogeneity in human epilepsy is a fundamental principle unifying the disease's etiology. So to start off, I want to motivate why the KBI generally, the Krembil Brain Institute, and why our group specifically is focused on epilepsy. And quite simply, it's because epilepsy is the most common serious neurological disorder in the world. Interestingly, we've been able to characterize this disease in some form or another, going back to the dawn of modern medicine, to the ancient Greeks and Hippocrates. And we've actually had decent pharmacological interventions to prevent seizures in patients with epilepsy for more than 100 years. But what's interesting about these pharmacological treatments is that they don't work for about 1/3 of patients. And the underlying hypothesis motivating these interventions has remained largely the same over the course of this century. So let's dig in a little bit more into these drugs. And you recognize that all of these treatments are motivated by the hypothesis that seizures arise from excess excitation. This makes intuitive sense because we can think of seizure as an overactive brain state. If you recall my presentation at Krembil Research Day back in the before times in 2019, I made an analogy between our brain and a car, and a seizure being like our car careening off the highway at high speeds, completely out of control. So under this excess excitation hypothesis, it makes sense that our pharmacological interventions would aim to increase inhibitory signaling via the neurotransmitter GABA, or decrease excitatory signaling by blocking the neurotransmitter glutamate. But again, these treatments only work for about 2/3 of patients with epilepsy. And if we want to design new treatments for the remaining population, we need new hypotheses underlying epilepsy, the disease, and seizure onset to motivate these treatments. Now, in parallel with this, our group has also noticed an emerging consensus in the literature that heterogeneity at a cellular level is not only a preserved feature throughout the body, but specifically serves a role in the brain. Neural heterogeneity facilitates information coding. Because this heterogeneity serves such an important physiological purpose, it makes sense that its disruption might yield pathology. And that disruption actually ends up being a unifying feature of what may seem at first glance to be a disparate group of causes underlying epilepsy. For instance, some epilepsies are caused by the specific loss of an individual cell type, which can be viewed as homogenizing the entire population through the loss of that diverse cell type. Even within particular cell types, misexpression of ion channels, another known cause of epilepsy, can homogenize the properties within that cell type. Finally, a process known as synaptic sprouting, which accompanies seizures and particularly those brought about by traumatic brain injury, can homogenize the inputs to these neurons. So all of these disparate causes of epilepsy can be recontextualized as losses in neural heterogeneity. But this hypothesis has yet to be directly studied in any significant fashion. So we wanted to approach this question, and we wanted to do so in a way that fully exploits the advantages of the Krembil Research Institute. One of those is our relationship with Toronto Western Hospital and the fact that my supervisor, Dr. Taufik Valiente, is himself a neurosurgeon. That gives us access to live human cortical tissue from his surgical resections that go directly into experiments performed by my amazing colleague, Dr. Homer Moradi Chameh, who characterizes the individual properties of single human neurons using patch clamp recording. It's worth emphasizing that at Krembil, we are one of literally a handful of labs in the world that are able to do this level of recording in human neurons. I'm lucky enough to be able to take this unique data and have it constrain computational models, and in turn, mathematical analyses, an area in which my other supervisor, Dr. Jeremy Lefebvre's expertise is particularly useful. And what I'll show towards the end of this talk is how we're able to close this experimental computational theoretical loop and use our theoretical insights to explain somewhat surprising experimental results. Now, what are those experimental results? We primarily wanted to characterize the excitability of our human neurons. We did so via a distance to threshold measure, which is simply the threshold potential of these neurons subtracted from its resting membrane potential. We measured this in 3 different populations: an epileptogenic population taken from the seizure-producing zone, and 2 different non-epileptogenic, non-seizure-producing zones. And what we notice is that the spread or the variability or the heterogeneity in this distance to threshold measure is significantly reduced in our epileptogenic population. This is more easily appreciable by these Gaussian fits of the data on the right. You see that the red curve has a much clearer, sharper peak in the data. The width of this curve is a way that we can quantify the heterogeneity of these populations, which mathematically we refer to as sigma, and that's how I'll refer to that heterogeneity throughout the rest of this talk. Now we take those experimentally derived sigma values and implement stochastically spiking neurons split into 2 populations, an excitatory pyramidal population and an inhibitory interneuronal population. These populations have both inter and intra connectivity. Now, when we look at these simulations with healthy levels of high heterogeneity, as we increase the drive to the system, we don't see a significant change in the population behavior. I've characterized that to the right. You can see this sort of population analog where there isn't a significant difference in the dynamics as the drive increases. We quantified that with measures over 10 independent simulations, the blue being the synchrony, and the black being the excitatory firing rate, the gray being the inhibitory firing rate, the fainter curves being plus or minus 1 standard deviation. And what you notice is that the increase in these measures is largely linear as we increase the drive to the system. That's contrasted severely with what we see when we have pathological low levels of heterogeneity. Early on in these simulations, we see a sudden onset of hypersynchronous, hyperactive dynamics. This is very similar to seizure onset because, again, seizure is a hyperactive, hypersynchronous brain state. So at minimum, this presents anecdotal evidence that decreased neural heterogeneity is permissive of the seizure state. We wanted to strengthen that connection, and this is where we brought our mathematical tools into play. I won't dive too deep into the mathematical rabbit hole given my 10-minute limitation here, but essentially what we're able to do with these mathematical tools is quantify an fundamental structure underlying these network dynamics. We do so using something called fixed point analysis. The primary difference between the high and low heterogeneity case is that in the high heterogeneity case, we always have a single fixed point. In the low heterogeneity case, early on, we will have multiple fixed points, 2 of which disappear. This behavior is known as a bifurcation. This is something that's been studied in the mathematical literature for centuries. And it's known that bifurcations underlie sudden changes in dynamics, very much like the sudden onset of hypersynchronous, hyperactive activity that we saw previously. So taken together, this allows us to say that epilepsy can be recontextualized as a progressive loss of biological heterogeneity. We've shown experimentally that this loss of heterogeneity exists in human epilepsy. We've shown computationally that a loss of heterogeneity yields dynamical changes associated with seizure onset. And we've shown mathematically that there's a fundamental explanation that connects this loss of heterogeneity to this change in dynamics. Now, here's the kicker. We found something very surprising during our experimental characterization of these neurons. Our epileptogenic population actually tended to be less excitable, often significantly so, than our non-epileptogenic population. You see that via these FI curves, repetitive firing frequency in the y-axis, input current to the neuron on the x-axis. Now, independently, when we characterized our models of neurons, we found that the population averaged input-output function is more flat for our blue population of neurons, that is our heterogeneous population. The dark curve here are the average input-output functions. The fainter curves behind are single units. This is an example of the well-known ability of heterogeneity to linearize these input-output relationships. And lo and behold, I can quite simply plop that FI curve right onto this mathematical characterization of the effects of heterogeneity. And it goes towards explaining the surprising difference. It in fact seems like our loss of heterogeneity in the epileptogenic population can explain why we saw these neurons were less excitable on the single neuron level in our experiments. This closes that experimental theoretical loop. We're using our mathematical theory to explain our experimental results. And it goes towards this new perspective that perhaps it is a loss of heterogeneity, of neural heterogeneity, that is the unifying feature of all these varying causes of epilepsy that we see, and may help to explain why excitability changes may not happen at the single neuron level, but may happen more at the network level. And with that, I'd like to thank my fantastic advisors and collaborators, and I'm happy to answer any questions you might have.
Okay, so I think we'll start with maybe Liang. Would you like to unmute yourself and make your comment with regard to Scott's presentation? Any other questions, put them on the chat function.
Yeah, it's very nice presentation and very elegant studies, which I really are excited about this. I would just say the broad sense at the beginning of introduction, maybe for people outside of the field, maybe indicating that the non-neuronal cell should be considered. Although the individual study can focus on the different type of neuronal cells, but overall, I mean, excellent study. Yeah.
Thank you.
Are there any other questions? I have a question. How would you adapt this concept to newer therapeutic approaches? Because you said that not everyone responds. And how do you envision this to help you define better therapies or newer therapies?
Yeah, so I think that the idea of trying to rescue neural heterogeneity as opposed to rescuing inhibitory signaling is something that can lead to new avenues for these type of interventions. For instance, the use of deep brain stimulation and neuromodulation, electrical neuromodulation to prevent seizure, you can sort of think about that as imparting more heterogeneity to the system. If you're using these devices to impart more noise or more randomness to the system, that is a way of imparting heterogeneity back into a system that perhaps has lost it. So I think, and this was actually some other work we did where we showed theoretically that using a noisy stimulus can actually have seizure preventing effects in sort of the in silico setting. That's something we're excited about. But again, I think the other avenue of this is that it can go towards pharmacological treatments that are focused less on either blocking this excitatory signaling or enhancing this inhibitory signaling, but instead finding a way to rescue this heterogeneity, whether it be rescuing the proper expression of these ion channels, rescuing these particular cell types from cell death, these are targets for these interventions that haven't been as well studied that I think could be ripe with interesting applications. Looks like Mary Pat.
We have a couple of other questions, but I think the problem is that we're running a little short of time and we need to move on. So if you wouldn't mind asking Scott later these questions, I think we'll move on to our next presentation.
Find me and gather. I'm happy to help out.
That was excellent. Thank you, Scott. So our next presentation this morning is from Akshay Gurdita, who works with Valerie Wallace. And I guess we're ready to go. Go ahead.
Hi, everyone. My name is Akshay Gurdita. I'm a PhD student in Valerie Wallace's lab, and I'll be presenting on some of my PhD project work, which is looking at rod photoreceptor somal migration during neuron retinal development. So first of all, the retina is this neural tissue at the back of the eye that's responsible for detecting light, converting these light signals into electrical responses that are then sent via the optic nerve to the brain. And within the retina specifically, we have rod and cone photoreceptors that are their outer segments or the parts of their cells that are actually responsible for detecting this light stimuli and then converting it into an electrical response, which is fed into the rest of the retinal circuit. And in diseases like retinitis pigmentosa or age-related macular degeneration, which involve the loss or degeneration of photoreceptors, this of course leads to vision loss and blindness. And there are no cures for either of these diseases. And so one hopeful therapeutic approach was cell replacement therapy, and specifically the idea of transplanting immature donor photoreceptors, with the idea being that if you subretinally inject immature donor photoreceptors, that these photoreceptors would then migrate into the host retina and develop and form mature outer segments and synapses so that they could functionally respond to light and then appropriately relay those signals. However, our lab and several other groups have demonstrated that the majority of cells do not exhibit this phenomenon. The great majority of cells actually just remain in the subretinal space and instead undergo cytoplasmic exchange with the host retina. And so if we want photoreceptor transplantation therapy to become a viable therapeutic strategy, then we need to better understand these processes that are required for true cell integration to actually occur. And of course, unfortunately, all three of these fields have significant gaps in our understanding. And so my project aims to look at rod photoreceptor migration, with the hope being that understanding how photoreceptors migrate may provide some insight towards improving the ability of transplanted photoreceptors to functionally integrate. So the conventional understanding about rod photoreceptor migration is that during retinogenesis, progenitor cells move from the basal side of the retina to the apical side of the retina, where they undergo mitosis and divide and produce photoreceptors. And photoreceptors are specifically born between E18.5 to P5 in mice, with a peak at P0. And these rod photoreceptors are born at the apical side and eventually, over time, develop this outer nuclear layer, which consists of several layers of rod somas. And the conventional understanding is that because rod photoreceptors essentially end up residing where they are born, they don't exhibit any obvious large migration pattern or behavior, and that all of the cells within the outer nuclear layer are all very homogeneous and the same in terms of the rods within the outer nuclear layer. However, interestingly in our lab, we noted that when we tried to label these rods using a subretinal injection of a lentivirus into P0CD1 mice, and then looked at these retinas at P14 when retinal development is largely complete, we found that instead of labeling rods throughout the outer nuclear layer, which, as I said before, the somas span the entire width of the outer nuclear layer, our lentiviral reporter only tagged the subpopulation of rods that sat on the basalmost side of the outer nuclear layer. And importantly, these rods appeared to be postmitotic at the time of infection because they were not labeled with EDU. And so we realized that we could use this lentiviral labeling of rod subpopulations to try and characterize rod migration in a way that was never done before. And we can use this to also ask several questions. For example, if rod somal positioning is dependent on rod birth date, and how these basal rods actually migrate to that final laminar sublaminar position. And we can also use this to ask if rod migration is altered in models of retinal degeneration. And so to answer the first question, what we did was, again, doing subretinal injections of lentivirus, but this time at P0, P3, or P5 Rosa MTMG pups. And the lentivirus is a rod-specific promoter driving Cre expression. So in Rosa MTMG pups, infected rods will end up expressing a membrane GFP. And so here's a summary slide of this experiment where you can see P0, P3, and P5 injected pups. And the lentiviral reporter, the membrane GFP, is in green. And what this data shows is that injection of lentivirus at later time points exhibits a greater apical basal distribution of rods within the outer nuclear layer, suggesting that rod somal position is actually dependent on the birth order. And if we just blow up those images here and show some quants alongside, what I'm trying to say here is that the distribution of rod somas within the outer nuclear layer becomes less pronounced over time. You get this sort of more equal distribution throughout the outer nuclear layer. And this is inversely correlated with the distribution of EDU positive cells within the outer nuclear layer. And this is consistent because we're essentially infecting more and more post-mitotic rods at later time points. And so, like I said before, going back to our P0 injected mice, where we see this basal layer of rods, we want to ask if these rods migrate to that final laminar position, how they do that. So again, by performing subretinal injections into Rosa MTMG pups with this rod-specific Cre virus, but doing a time course analysis at several time points, several days post-injection, leading up to that P14 time point when we see that basal rod distribution. And here's a summary of this experiment, looking at different time points of days post-injection leading up to that P14 time point when we see these rods at the basalmost part of the outer nuclear layer. And what we can see if we look at the magnified region, relative to the position of the outer limiting membrane, which is the apical most region of the outer nuclear layer, you can see that rod somas actually appear quite close to the apical margin early on in development. And over time, these rods actually slowly migrate basally to their final sublaminar position within the outer nuclear layer by about P10. And the quantitative analysis correlates with these images as well. And so for the last question, to ask if rod migration is altered in models of retinal degeneration, again, we lenti-infected P0 pups, but this time we used transgenic models that are often used to study photoreceptor degeneration. And again, harvesting at P14 and comparing to the wild type basal laminar position that we would normally see in wild types. And this is a summary slide of this experiment where what you can see is that on the left is the wild type and on the right is the CRX knockout. On the left is the wild type and on the right is the degenerative model for each of the three degenerative contexts. And the lentiviral reporter again is in green. And if you look at the magnified region, what I want to point out is you can see the altered distribution in models of degeneration like the CRX knockout or the NRL knockout. The photoreceptors here are spread throughout the outer nuclear layer compared to the wild type, which exhibit that basal distribution. And in cases like the RD1, it appears as though there are fewer lenti positive cells or rod photoreceptors at the basal margin compared to the wild type context. All of this to simply say that rod migration at least appears altered in models of degeneration and may in fact be implicated in the degenerative phenotype as well. And so I want to point your attention to that previous model I showed at the beginning of the presentation of rod photoreceptor development and contrast that with sort of an updated picture based on some of the data I've showed here today, where rod photoreceptors actually do exhibit a basal migration pattern over time, where they reach the final sublaminar position, and their final sublaminar position actually appears to be dependent on birth order within the retina. However, this data doesn't actually say anything regarding the mechanism of migration, whether it may be a non-cell autonomous mechanism at play. For example, as progenitor cells migrate apically, they could be passively displacing basal photoreceptors, or there could be a cell autonomous mechanism at play intrinsic to postmitotic rods driving that basal migration. And so some of our future experiments aim to try and tease this apart by targeting subpopulations of rod at the same time using electroporation and lentivirus. And by being able to target distinct subpopulations, using these two methods and either manipulating one or the other, we can see if manipulation of one group ends up targeting or affecting the other's ability to migrate or their positions within the outer nuclear layer by the end of development. And with that, of course, I'd like to thank everyone in the lab and all of our funding sources, and I'd be happy to take questions.
Great, thank you. So our first question is from, let's see, Leanne Zhang. Do you want to ask your question?
Yeah, quite interesting. I'm wondering, did you examine the viral positive cell after postnatal 14? I think the POP just opened the eye around the 10 days. So
yeah.
I'm wondering, can the positive cells still show afterwards?
Yeah, I've done injections up to P21, but I just didn't show data here. And the basal migration, the cells are pretty stable still at P21 even. I mean, there's plans to go even beyond that into sort of the conventional adulthood as well. But in terms of the major retinal development is done by P14. And so that's why, just for the sake of the time course of the presentation, I showed data up to P14. But we have gone up further to P21 and we do have plans to go further. I don't expect, because this is a Cre virus, once the cell has the Cre and is expressing the reporter, it should remain. It's not like the reporter would disappear or anything later on.
Thank you.
Michael, do you want to ask your question?
Sure. Good job, Akshay. Thanks for this nice presentation. One question. So you have a mispositioning of the rods. My question is it due to impaired migration, actually, or is it just due to some sort of reorganization of the cells that are left there? Because if I remember correctly, those mutants have, those cells are dying in those mutants.
Yeah, right. So in terms of the repositioning after death, actually, in the case of the CRX knockout and the NRL knockout, degeneration doesn't quite begin yet at P14. CRX knockout, I believe the degeneration begins at about P21. And for the NRL knockout, I believe it's also P21. So in those models in particular, loss of cells wouldn't necessarily be responsible for that repositioning. And just simply looking at the outer nuclear layer thickness too, that is apparent that there hasn't been a huge reduction in the loss of cells yet. With the RD1 though, that is possible. But in the case of the RD1, we still see that basal pattern, just a reduced number of cells, because potentially the likely answer is that yes, because of thinning already beginning in the RD1 so early, that could be accounting for a loss of the number of infected cells there. But that brings another question on as to if basal photoreceptors that are being labeled are dying first over apical ones, and that's something that we're interested in asking.
Okay, thank you.
Jeremy, would you like to ask your question?
Sure. Great talk, actually. Really interesting stuff. I was sort of struck, it's kind of a follow-on from Michael's question, which is sort of thinking about, I guess, controls for what this is, that this really is a migration phenotype that you're studying. And at the beginning of your talk, you mentioned sort of how important labeling or sort of understanding what your labeling really is, what you're actually studying and what you're counting. I'm wondering whether there's any possibility that the transduction, the lentivirus with the reporter, is actually creating a subtly altered population of cells, and that these differential locations might be essentially a subtype of photoreceptor that you've kind of created by doing that, and if there's a way to control for that possibility.
So something I didn't show in that summary slide, because of the 3 different degenerative contexts, I used 3 different lentiviruses in each case as well. And so, for example, in the beginning, I showed a rod-specific lentivirus I was using as a reporter. In the CRX knockout and the NRL knockout, I'm actually not using a rod-specific promoter because CRX and NRL are not really there, so you can't really say that rods would express the same thing. In the wild types, though, I'm using the exact same lentivirus. So in terms of the cell I'm labeling, The lentivirus should be labeling, I mean, in the wild type, the same group of cells as the CRX knockout because it's the non-cell specific promoter, it's just a ubiquitous promoter. And that brings up the question then of, well, okay, if they're not really photoreceptors anymore because they lack CRX, what are they? The interesting fact about that is, well, they still form an outer nuclear layer, but the outer nuclear layer or the pattern within that outer nuclear layer is different from a wild type context. So it still sort of begs the question of, well, what are these cells if they're not true photoreceptors? The point being, these cells that exist in the outer nuclear layer don't follow the same distribution pattern in a wild type context. So something is still different there between the two that's still interesting to go after.
Okay, thanks.
Thank you very much for an excellent talk. Unfortunately, I forgot to unmute myself. Now we're going to move on to Jihan Nasri, who is working with Vinod Chandran. Jihan?
Good morning, everyone, and welcome to another session. I believe the last session of this morning's oral presentation. My name is Jihan Nasri. I am a second-year master's student in the Glatman Chandran Lab, and today I will be presenting, oh, sorry, am I on mute?
No, we can hear you.
Okay. Sorry, I'll start my videos. Yeah, sorry. So again, sorry, my name is Jihan Nasri. I'm a second-year master's student in the Glatman Chandran Lab, and today I present my project entitled Screening Anti-Inflammatory Properties of HDAC Inhibitors for Psoriatic Disease. So very briefly, psoriasis is an immune-mediated inflammatory skin disease characterized by scaly plaques of the skin. It affects up to 2 to 3% of the world's total population. And out of those affected, 30% of psoriasis patients will also have an inflammatory arthritis known as psoriatic arthritis or PSA. I really like the Caspar definition of PSA, which is established inflammatory articular disease with at least 3 points from the following, either current psoriasis or a history of psoriasis, dactylitis, just articular new bone formation, rheumatoid factor negativity, and nail dystrophy. Together, psoriasis and PSA represent a clinical and immunopathogenic continuum known as the psoriatic disease. And going forward, you'll hear me use these terms accordingly to refer to either or both diseases. So in terms of pathogenesis at present, while the exact cause of psoriasis and PSA are not known, it is agreed that both genetic and environmental factors play a role in activating psoriasis. So when psoriasis is triggered either genetically or environmentally, or even due to the loss of self-immune tolerance, what happens is that there is an aberrant activation of both innate and adaptive immune responses to support the initiation as well as subsequent maintenance of psoriatic inflammation. It usually begins here with the activation and maturation of dendritic cells in the epidermis of the skin and secretion of various factors such as interleukins 12 and 23 to stimulate T helper cells. These T helper cells will then secrete more pro-inflammatory factors, cytokines, and chemokines, finally resulting in epidermal keratinocyte hyperproliferation, in other words, psoriasis. From here, the development of psoriatic arthritis is not yet a complete story. However, when patients do develop PSA, many more immune pathways and players are involved to form this severe inflammation that is highly associated with T cell subpopulations as well as other immune subpopulations like dendritic cells, macrophages, neutrophils, and natural killer cells. These mediators of inflammation will then secrete even more pro-inflammatory cytokines to activate resident cells as well as joint-specific cells, resulting in the recruitment of immune cells to the joint, creating a highly inflamed synovial microenvironment, which then subsequently leads to the formation of abnormal synovial tissue growth, enthesitis, and eventually joint damage, all features of PSA. So as an implication of having this severe inflammation in psoriatic disease, either psoriasis, PSA, or both, patients have reported a reduced quality of life, both in terms of physical and mental health. And unfortunately, despite the availability of current treatments like DMARDs and biologics, there is still an absence of a long-term effective treatment for psoriatic disease, with many patients still reporting persistent inflammation, as well as difficulties in achieving clear or at least clear skin. Fortunately, a class of small molecules called histone deacetylase inhibitors or HDAC inhibitors has been proposed as a new promising anti-inflammatory treatment for psoriatic disease. As their name suggests, HDAC inhibitors, for example, here I have SAHA or varinostat, they function to inhibit or block the activity of HDAC enzyme proteins. And at present, there are two classes of these HDAC inhibitors, the non-selective pan inhibitors that would target either a specific class of HDACs or like class 1 or class 2, or a combination of HDAC protein from these different classes listed here. And then we have the isoform selective HDAC inhibitors that selectively targets a specific isoform of these HDAC proteins. So for example, HDAC1 or HDAC6 proteins respectively. In terms of why these inhibitors have been advocated for use, actually not just in psoriatic disease, but also other immune and inflammatory diseases, is due to a number of reasons depending on the disease. But for psoriatic disease, HDAC inhibitors may be useful, especially as anti-inflammatory agents, since both psoriasis and PSA have been linked to epigenetic modifications, and that too, HDAC inhibitors seem to display anti-inflammatory abilities in many preclinical studies. However, there are limitations to this rationalization, as well as gaps that need to be further addressed. Among others, most of the HDAC inhibitors that were used in those clinical studies are so far non-selective pan inhibitors. And we know from previous cancer studies that these types of inhibitors often cause off-target effects. So then in terms of selective HDAC targeting, unfortunately, the anti-inflammatory value of isoform selective HDAC inhibitors rather than non-selective pan inhibitors has yet to be determined. And overall, the use of these of any HDAC inhibitors for the purpose of treating inflammation, especially for the skin, is relatively unexplored. So in order to try and fill in the gaps that were identified, we hypothesized that selective HDAC inhibition in keratinocytes by isoform selective HDAC inhibitors will exert effective anti-inflammatory effects at non-cytotoxic concentrations. To do this, I evaluate the in vitro anti-inflammatory efficacy of 26 selective HDAC inhibitors by 1, inferring for their cytotoxicity levels, as well as 2, assessing their ability to suppress pro-inflammatory cytokine expressions. And I performed these assays in cells that closely represent psoriatic disease, so immortalized human Hakat keratinocytes. And what we found was that by using the cell titer blue cytotoxicity assay, plotting for cell viability against concentration, overall, our panel of HDAC inhibitors displayed varying levels of cytotoxic activity in the Hakat cell line, as seen by some representative compounds here in table 1. Favorably, most of our isoform selective inhibitors were found to be less cytotoxic than the pan HDAC inhibitors, tracostatin A and SAHA, shaded gray right here. And you can tell this by the higher IC50 values of our selective compounds compared to the shaded compounds. And this is ideal, as it means that more selective inhibitors are actually needed compared to pan inhibitors to kill the same amount of cells, widening our therapeutic window. So then in terms of this compound's actual anti-inflammatory ability to suppress pro-inflammatory cytokine expressions when Hakat cells were grown to confluency and then pre-incubated with compounds before stimulation with TNF-alpha, it was found by Luminex multiplex of the cell culture supernatant that from the 26 selective inhibitors tested, 9 showed sufficient anti-inflammatory ability to significantly reduce IP10 production at non-cytotoxic concentrations, as seen from these representative graphs in figure 1 and then summarized in table 2. And I'll talk a little bit more about how this data was analyzed. Looking at the cell viability graph here on the left, or alternatively here, when compounds were tested and at different concentrations. So here I tested 100 nanomolar, 1 micromolar, 10 micromolar, and 50 micromolar. The amount of viable cells were measured first and screened for the effective concentration. And what I mean by effective concentration is the concentration of compounds at which the amount of cells is at least above 40%. And then using those effective non-cytotoxic concentrations, the cell culture supernatants at those concentrations were measured for the production of, here I have IP10, and what we're looking for is a reduction of IP10 expression. And you can see that for SAHA at 1 micromolar here and MMH 172 at 10 micromolar. And then the rest is summarized in table 2. However, I will say that the anti-inflammatory effects displayed by these compounds here were seen at a rather high concentrations of around 10 to 15 micromolar. And unfortunately, these selective HDAC inhibitors are known to be isoform selective only at lower concentrations, commonly below 1 micromolar. Hence, the anti-inflammatory effects displayed here were most likely due to non-selective HDAC inhibition, similar to the effects displayed by PAN inhibitors. But more studies are needed to further understand this. And so what remains to be done for this project in terms of future directions are to complete the same assay, but in a different yet still relevant cells, the THP1 macrophage cells, then to validate the efficacy of these compounds in a more sophisticated disease models, and then lastly to investigate the anti-inflammatory effect of HDAC inhibition. And so to conclude, HDAC inhibitors are a class of anti-cancer small molecules that have been proposed as a new promising treatment for psoriatic disease. However, the use of these inhibitors for treating inflammation, especially for the skin, is not yet demonstrated. Here we present the first ever study demonstrating the efficacy of selective HDAC inhibitors as anti-inflammatory agents in keratinocyte cells. And we found that from 26 selective inhibitors tested, 9 showed sufficient anti-inflammatory ability to significantly reduce IP10 production at non-cytotoxic concentrations. However, these concentrations were higher than the concentration at which these compounds are known to be selective. And so more studies are needed to further understand these effects, and the use of HDAC inhibitors as anti-inflammatory agents in other psoriatic disease models warrants further investigation. With that, I would like to thank members of the Gladman Chandran Lab, as well as the Gunning Group at UTM, and everyone in the audience for listening. As well, I'll be happy to take any questions if there are any.
Okay. There are no questions yet. I do have a question. Could you tell me, I mean, you focused on keratinocytes. What exactly is the role of keratinocytes in the inflammatory process? Like what particular cytokines did you focus on beyond IP10?
So I didn't show in my slides, we did test a couple of other cytokines, IL-6 as well as IL-8, which are a little bit more relevant in keratinocytes, but unfortunately, those cytokines, as we go through the assay, the cytokines became less manipulable under our conditions. And so IP10 was the only one that worked. But in terms of keratinocytes, it does play a huge role in psoriasis. And as I mentioned in the pathogenesis, I think I can still show it here. Yeah, over here it's a big part of the pathway, and so it's, I guess, as a point of measurement for us.
Mm-hmm. So, I mean, does it play a role in recruiting dendritic cells, or what precisely role do the keratinocytes play?
Right. Sorry. Thanks for the question. So it's more of it's more of an endpoint. Once you have all this inflammation going around, you know, from the dendritic cells, from the T cells, keratinocyte proliferation is what happens as a result of that, and it's something that you can see in patients. So for us, is a point of measurement and as an endpoint measurement.
So you mentioned, because I'm still waiting for questions, you mentioned that you were next going to look at macrophages, and why did you pick that particular cell type? What role do macrophages actually play in the process?
Right. So there were a lot of cells that we initially planned for this project. We looked at T cells as well. As well as we did consider for a while neutrophils, but in the end, it was macrophages because compared to all the other cells, macrophages are more interesting in a sense that it wasn't tested as much for this indication, and we thought that would be interesting. But again, in terms of PSA, in terms of the inflammation in PSA, macrophages do play a huge role in that pathway. It was a way for us to kind of capture PSA as well.
Okay, great, thank you. So I think that concludes our talks for this morning. I would now like to invite people to exit Zoom and then enter into GatherTown, where you can then attend the poster sessions. Great talks, everyone, and we'll re-adjourn for more talks after the poster sessions.
Hi, everybody. Since it's 1 o'clock, we'll get started with our second set of oral presentations. Hopefully everybody had a great experience in the poster rooms. And hopefully everybody can manage to find their way in and not get blocked at the entranceways. So thanks very much. And we'll do very much like as Joan had done earlier. So if you have a question, just type in the chat that you have a question, and I'll invite you to unmute yourself. I'll remind the presenters that the presentations are 10 minutes long, and then we have 5 minutes for questions. So having said enough to get a few more people in the door, then we'll get started. Our first presenter is going to be Nadim Murtaza from the Karan Singh Lab. So take it away, Nadim.
Okay, I'll just get started. So as Mary Pat said, I am a PhD student in Karan Singh's lab. And today I'm going to be presenting on a neuronal platform to identify shared neurodevelopmental disorder signaling networks and the applications that we're using it for. So just to start off, our lab studies neurodevelopmental disorders, and more specifically, we have a large focus on autism spectrum disorder. And so autism spectrum disorder is a broad range of neurodevelopmental disorders with varying severities between individuals. It has 2 core symptoms: impaired social communications and repetitive behaviors. And in the last study by the Public Health Agency, they found that 1 in 66 children are actually diagnosed with ASD. Now, although there's some pharmacological treatments and behavioral therapies, these aren't directed to the core symptoms. And so there's not really a robust treatment for the disorder. And all of this is just due to the lack of knowledge in the underlying pathophysiology. Now, in the last decade, there have been multiple whole genome, whole exome sequencing studies that have looked to identify mutations in individuals with ASD. And because of this, we have a large list, more than 500 genes that are associated with ASD in some manner. And one of the issues that has come up is we don't really understand how all these genes work together. In fact, if you try to look up the interactions or association between these genes using databases such as the string database, you see this kind of output where, yes, there's a lot of genes that are connected together and so have some sort of association, but the majority of them actually don't have any known interaction with each other. And so even if you did some sort of pathway enrichment, you get broad brain cell function interactions. And overall, a lot of these issues is because a lot of these connections are based on RNA expression. So they don't really show interaction at the protein levels. And even if they do, these experiments aren't actually done in neurons, which, because this is a brain disorder, the interactions that we might see might not be relevant to the disease. And so the real question that has been really taking place now is, how are these ASD-associated risk genes functionally connected? And to answer this question, we decided to use a system known as BioID. And the way BioID works is essentially you have a BRA ligase, which is a promiscuous biotin ligase that biotinylates anything around it within a certain range. And so by actually fusing this protein to your protein of interest with a linker, for example, you can essentially biotinylate anything around your protein of interest, whether it's close proximity or interacting directly. And so this is important because this really captures strong and weakened transient interactions. And so strong interactions, such as the ones you would normally get through immunoprecipitation, but the weakened transient interactions that you can't normally get through IPs, you can really get through BioID, because the way the system works is that that protein just has to be around long enough to be biotinylated, and then it can move on to its next target, but you can go back and find it afterwards. And so because of this, you typically do get more hits in immunoprecipitation. Most of these new hits are weak and transient interactions. And so traditionally, bioID has been used primarily in cell lines for mapping compartments, and there's large studies out there that have shown proteins mapped to different compartments. But in our case, we really wanted to utilize this in a more neuron-specific primary cell line model, because we really wanted to understand the interactions that are more specific to neuronal function, since ASD is a brain disorder. And so the system that we created utilizes mouse coracle neurons with a glial co-culture. Just to give a brief overview of it, we use lentiviral infection to overexpress these bioID fusion constructs in these mouse neurons. We wait a few days and then we allow biotinylation by adding biotin to the cultures. And after 18 hours, we lyse these cells, we pull down the biotinylated proteins, and through the whole processing protocols, we finally run them through the mass spec and identify whatever proteins are interacting with your protein of interest. And so just to show you some validation, we use PC95, which is a well-known postsynaptic density protein, and in fact, it actually is a high-risk ASD gene as well. Here you'll see that there's some staining of infected neurons, which show GFP and MAP2, and you really see that the localization of PC95 compared to luciferase control shows a more punctate localization, which you can see in this close-up image of the dendrite, where you see PC95 is very punctate, so most likely in synapses, while luciferase is more just diffuse within the dendrite. And this makes sense because luciferase doesn't have a known localization in neurons. It's not normally expressed in mouse neurons. And so after we decided to do bioID on PS95, these are the proteins that we identified. And here I'm just showing you the reactome pathways are enriched in this data set. And you really see that most of these proteins are synaptic proteins, so AMPA receptors and NMDA receptors, and also scaffolding proteins such as DIL-GAPs, DLGs, homers, and shanks. So a lot of the proteins that you really do expect to be within close proximity or directly interacting with PS95. And so just comparing this to other studies, so there's been a study that's done in vivo bioID of PS95 in mouse brains, and also studies that have done IPs in PS95 in mouse brains as well, using mass spectrometry. And so here you see there is a core 25 proteins that seem to be shared within all of these studies, including ours. And this really just shows all of these synaptic and scaffolding proteins that we found. Now, the real important part of this was that when we compared it to HEC293 cells that we did PS95 bioID on, you really see that all these synaptic proteins and scaffolding proteins aren't really in these cells. And this just drives home the fact that, yes, you can use HEC cells to do cell line compartment mapping, but if you really want to find disease-relevant protein interactions, you do have to use a disease-relevant cell type. And so we've done bioID on 39 ASD risk genes. If you look at the interactions that are known between these genes right now, you really see that there's not much. There's interaction between the synaptic proteins, which have widely studied, but the rest of the 39 proteins that we chose based on the wide range of functionality don't really have any connectivity. However, when we take all of the different bioIDs that we did and we combine them into a shared network, this is a network that we get. And so just to explain this, the dark nodes are the 39 ASD risk genes that we did bioID for, and the smaller nodes are the proteins that were identified through the bioID process. And so here you see a lot of color-coded nodes, and the basic thing that you can tell is that a lot of these proteins were identified multiple times in different ASD risk gene bioIDs, meaning that they're shared between these genes. And so now you can really see that this bioID system really shows you the connectivity between these ASD risk genes, not directly, but through other interactors. And what was even more surprising is that even though we started with 39 ASD risk genes for the bioID process, we actually identified 120 ASD risk genes within this network of 1,000 proteins, which you can see with the red highlighted nodes, which might be a little bit hard to see on the screen. But this is really important because although it's already been implicated that yes, ASD risk genes do work together, this really shows that on a protein and interaction level, these ASD risk genes probably have common pathways. And so when we actually look at the common pathways that are enriched in this shared network, using the reactome pathway gene sets, you really see that some of the most enriched pathways are synaptic pathways, which makes sense. And even things like polarity and Wnt pathways, which also makes sense. These are heavily studied pathways involved in neurodevelopment disorders. But what was interesting, we also got highly significant enrichment of pyruvate metabolism and TCA cycle proteins. And this is important because even more recently, there's been a lot of studies showing that individuals with ASD have mitochondrial dysfunction or mitochondrial disorders, or even monogenic syndromic forms of ASD also have deficits in mitochondrial function. So this seems to be a pathway that's heavily involved in ASD, and this is something we wanted to follow forward. And so going forward, one of the questions we wanted to ask was, can we identify novel pathways? And in fact, when we studied TAUK2, which is a gene, a high-risk ASD risk gene that we've studied extensively within our lab, we actually identified that yes, TAUK2, which we already knew to interact with synaptic proteins and neural projection proteins, which we previously shown, also interacts with some novel pathways that we really didn't know about in regards to TAUK2, such as translation and cellular respiration, which goes back to the pathways that we saw are shared between many different ASD risk genes. And so in fact, when we looked at mouse cortical neurons or even human neurons that we use CRISPR-Cas9 to completely knock out TAUK2 or introduce a patient mutation or individual that had ASD, which had the A135P mutation, you really see that when we use the seahorse assay, you see a significant increase in maximal respiration in both the heterozygous mouse neurons and the complete knockout of TAUK2 in the human iPSC and derived neurons as well. And when we look closer, we actually do see that in the human neurons, when we essentially stain for mitochondria through overexpression of MITO7 DS-RED, you see that the mitochondria in the knockout and the A135P mutants seem to be larger than the wild type. And so this really shows us that there is a mitochondrial dysfunction phenotype occurring within these neurons. And in fact, when we use proteomic analysis of TAUK2 knockout mice brain fractions, we really see that mitochondrial proteins are changed or downregulated in these mouse brains. So this really just drives home the fact that mitochondrial function is affected in these mice and is possibly regulated by TAUK2. And so one of the last questions we wanted to ask was then, now that we know these A135P neurons are affected in terms of mitochondrial function, can we use this system to also study other patient-derived mutations in different ASD risk genes? And so we chose a few different ASD risk genes, a few high-risk ASD risk genes, one of them being the GRIA1 protein, which is a AMPA receptor subunit, and also the PPP2R5B protein, which is a phosphatase. And so what we did is we decided to use different mutations that are either in different domains or spread throughout the protein. And you can really see that depending on what type of mutation or where the mutation is, we really do get a differential effect on the interaction, where larger circles mean an increase in interaction and a smaller circle means that there's a decrease in interaction. You can really see this difference with the GRIA1 mutations. And so we did the same thing with the A135P patient-derived mutation, and you really see that most of the interactions for TAUK2 are completely lost or reduced. And we already knew that TAUK2 mostly interacts with synaptic proteins. And so therefore we expected that there's going to be a decrease in synaptic functionality, which we actually did see as well. And here I'm showing you just human neurons stained for synapsin 1. And you really see that in the knockout neurons or the A135P neurons, you do see either a change in the synaptic number or the size of these puncta, which signifies there's some sort of alteration in synaptic formation. And even if you look at electrophysiology, you can really see that the frequency of spontaneous EPSCs are significantly reduced. So this really does show that the mutation dramatically affects synaptic function. And so just to conclude, what we've shown with our platform is that we can identify interaction networks for ASD risk genes that have known and unknown neuronal functions, and even share protein interactors between multiple ASD risk genes. We've shown that even though we started with 39 ASD risk genes, we ended up with 120, which really shows that ASD risk genes do work at the protein interaction level. And we showed that we can identify novel pathways, one of which was the mitochondrial function pathways in the TAUK2 neurons. And this is important because this seems to be a pathway that's been shared between multiple ASD risk genes. We showed that we can identify impacts of disease-associated mutations using bioID. And then finally, the real conclusion is that this ASD risk gene network identifies shared functional pathways between groups of ASD risk genes that can be used to categorize these genes and then identify possible common therapeutic targets for different subsets of ASD. And this is something that we are working to do and create this database. And so just to end off, there's some future things that we're trying to do as well. Because most of the screen was done in mouse cortical neurons, we're really trying to see, can we study and identify the networks in human neurons? And so we're utilizing cortical organoids, where we created a DOX-inducible system that lets us express the bioID constructs in a neuron-specific manner or any cell type-specific manner. And so far, this is what we've done, where I've created a DOX-inducible system, where if I add DOX, you start seeing expression of the bioID construct, which you can see with the GFP expression which looks like they're expressed around these rosettes within these organoids. And so finally, I just want to thank my lab and the different collaborators that have helped us throughout this project. Thank you.
Okay, great work. Thank you very much. I think we have time for only one quick question. Liang, you've got the floor.
Yeah, so he asked if biotinol proteins included, so that is all like membrane and nuclear proteins. So it does. Nuclear proteins are actually hard. In our case, we focus heavily on the neural communication or intracellular signaling proteins, because nuclear proteins are actually really hard to do bio-ID for. There's a lot of biotinylation just naturally in the nucleus, especially on histones. So we focus heavily on proteins that weren't nuclear. And in terms of identifying membrane proteins, bio-ID can identify membrane proteins. It does depend if the protein has an intracellular domain, or if that domain can be actually biotinylated, because biotinylation only occurs in the lysine residue, for example. And in regards to can this approach be used for brain tissue of adult mice? So the in vivo bio-ID study that was published back in 2016, they did it in adult mice, which they injected with biotin for about 7 days. So it is possible to do this system in brain tissue as well as adult animals.
Thank you.
Thanks.
Yeah.
Oh, thanks very much. Okay, wonderful work. Thank you so much for sharing it with us, Nadeem.
Yeah, thank you.
Okay, next up is Aranan Selvarajah from Danielle Andrade's lab.
Hi everyone, welcome. My name is Arunan Selvarajah. I'm a second-year master's student in Dr. Andrade's lab studying Dravet syndrome, and today I'll be giving a talk on an evaluation of adaptive behaviors, gait, and Parkinsonian features in adults with Dravet syndrome. So to start off, what is Dravet syndrome? It was first discovered in 1978 by Dr. Charlotte Dravet, a severe myoclonic epilepsy in infancy. It is a rare, intractable epileptic encephalopathy that begins in the first year of life. Approximately 80% of these patients have a SCN1A pathogenic variant, and you can see in this diagram below the spectrum of SCN1A disorders that they fall on the severe end here. And seizures are not restricted in childhood and continue into adulthood. And so SCN1A is expressed in several parts of the brain. This includes the cortex, the hippocampus, the basal ganglia, cerebellum, as well as the peripheral nerves. SCN1A translates for NAV 1.1, which is a sodium channel. And disruption in this channel results in intractable epilepsy, sleep, appetite, and motor disturbances. And these patients also experience a range of intellectual disability. And so in adults, Dravet syndrome is not well studied. Overall, we do see that there's a decrease in seizure frequency, and the types also change as well. So for example, the myoclonic and febrile seizures, they decrease, and the nocturnal generalized tonic-clonic increase. In the clinic, we also see that they do have some motor and cognitive deterioration, and parents of these children often complain about losing previously acquired skills. And in the clinic, but also in the literature, we do observe a crouch gait. And so in 2014, with the help of the Movement Disorder Lab, our lab showed that Dravet adults develop early signs of Parkinsonism in their early 20s. However, it's unclear if these Parkinsonian manifestations seen in our patients are a part of an ongoing neurodegenerative process. And so we wanted to determine if the gait, Parkinsonian features, and adaptive behaviors get worse over time. And we assess this through 3 assessments: the gait mat analysis, a modified unified Parkinson's rating disease scale scores, as well as the Vineland's adaptive behavior scales. I'll go into detail in each of these in a second. And so to be eligible, these patients attended our adult clinic, and they were, of course, 18 plus years old, not on antipsychotic medications, and must have had a clinical and genetic diagnosis. We analyzed their gait cross-sectionally in their most recent visit, whether it was 2019 or 2020. And then we also looked from a longitudinal perspective, their Vineland's and the MUPDR scores at 2 time points. And so in this gait analysis, this gait mat has sensors that are built in, and with a camera at the coronal and sagittal views, along with the protokinetic software, it's able to collect gait features that are listed here on the left here. So for example, the gait velocity, the cadence, the single double time ratio, which is the time on 1 foot versus on 2 feet in your gait, And basically they would be walking back and forth a couple of times here. And the gait variability index is accounting for the arm swing, the leg swing, and all the features as well. And so 21 patients were able to complete the gait analysis and the MUPDRS scores. The average age of our group here was 32. Male to female ratio is 3 to 4, and we just broke down our pathogenic variants here. And so what we were able to see, so this red dot here is the average 62-year-old group here. And based on these graphs, we were able to see that there's a Parkinsonian-like gait. And in Parkinson's, the cadence is consistent across all ages. So here along the x-axis, we have the age, and then on the y-axis, different gait features. And it's also similar in the stride width. It's pretty consistent as well. In Parkinson's, they do have an increase in gait velocity in our group here, and that's due to compensating for the balance issues. Again, you can see that here with the single double time ratio. So there we see a decreasing trend with age, and that means that as they're aging, they're spending more time on their 2 feet in their gait. Again, the stride length is decreasing with age. You can see that the 62-year-old average healthy group is performing much better. And again, the gait variability here is increasing with age. And the next slide, I again just want to briefly show these were the other parameters. Again, the 62-year-old healthy control is performing better than even the young Draves here. So here they're doing more or less the same in terms of the young Draves. For the ambulation time, the double support here, again, they're performing much better. The swing and the stance percentages, they go hand in hand. And again, 62-year-old is doing much better than the young Draves here. Okay, and so for the MUPDR scores, basically, this is a scale that assesses out of 4, and 4 being very Parkinsonian-like. And so we assessed these Parkinsonisms, so the left and right resting tremors, facial expression arising from a chair, gait, posture, and body bradykinesia. And what we see here is that these Parkinsonisms have a trend of getting worse with age. The more stronger trends here are the body bradykinesia on the right here, as well as the gait, which is of our keen interest. And then the total here, again, is a sum of all of the Parkinsonisms that I just mentioned. And again, we can see that they are increasing at a statistically significant rate here. From a longitudinal perspective, so on the left-hand side here, we have the patient number, the age at assessment, so both time points, and then the Parkinsonisms listed here. And we can see that 5 out of 6 of these patients have gotten worse. And then the one patient, patient 3, who didn't, is on Cinemet, which is levodopa. And that can help on it's assigned on a case-by-case basis, but can help with motor abnormalities. And in terms of the Vineland, so this is an adaptive behavior questionnaire, and it's adapted for intellectual disability and assesses 4 domains: communication, daily living skills, socialization, as well as the motor skills. And then it also assesses problem behaviors as depicted by maladaptive behavior scores. And so here again, so we only had 9 participants able to fill both questionnaires at both time points, and the average age here again is 32. Their age of onset of seizures is 6 to 7 months here. And again, I just wanted to point this out here. Just like in the literature, we see that they're febrile seizures and their myoclonic seizures are present in their first 5 years. And then in adulthood, they completely disappear and the nocturnal arises. And the interesting finding is their age of diagnosis is 23 years roughly. And so in terms of statistical analysis, we performed the Wilcoxon sign rank test, which is a non-parametric test to compare 2 related samples. And basically, although we saw decreases in several domains, they were statistically insignificant. That's partly due to the flooring effect, which basically means that they had minimal scores at both time points. And so to assess a difference is very complicated, and as well as a small sample size. So we couldn't stratify the data even further here. So what we did then was we compared the change in their raw scores along the subdomains. And so what we were able to see is that the expressive skills here, they improved, as well as the socialization skills here. So that's the interpersonal relationship, the play and leisure time, the coping skills, and we can see that those all improved. And the gross motor scores, they were either stabilized or got worse in several participants. And then the problem behaviors, they were dropped as well. So in terms of our discussion, thus far in the literature, this is the largest gait analysis in adults of Dravet. Again, we just see that these features and Parkinson's more or less get worse with age. The motor scores that we saw decrease in Vineland's, we also saw in the clinic. The flooring effect that I just mentioned, it means that it's not an ideal assessment for those individuals. They would probably need some sort of other assessment. In terms of conservation of their socialization skills. So there's actually a paper out there that shows that in children, they also saw that with the same questionnaire, they also saw some conservation of socialization skills. There's also potential habituation for neurodegeneration. So 5 of these 9 participants that filled in the questionnaire, they live at home. So they might not notice the day-to-day differences. And finally, the early evaluations such as these allow for a holistic approach to care and support services. And I just want to thank everybody here, as well as our funding sources, the Dravet Canada and Dravet Syndrome Foundation. Questions?
Great, thanks. I do see some questions in the chat. I'm actually going to start with Luca. If you want to unmute.
Thanks. Great talk. Just kind of curious. I mean, totally naive question from my end, but I would imagine like gait declines in a healthy individual with age as well. So you'd see sort of like similar relationships. So I'd be curious to know whether there's like a difference in the gradient in age compared to gait, you know, how effective their gait is between these individuals and healthy controls. And if you've done any healthy control studies and compared the two.
Great question. We're actually doing a second phase to the study right now, and we're matching healthy age match controls as well. And so we're hoping to see the thing that, to see that difference in that gradient there.
Great, thank you.
So we also have a question from Scott, and I think it kind of picks up on the question as well that was asked earlier by Liang. So maybe Scott, you can ask your question.
Yeah, so I'm curious, you've evaluated your patients with Dravet syndrome under this sort of Parkinsonian lens. Do you think that means that Dravet syndrome is acting on the brain similar to Parkinson's, or is there a sort of alternative pathway that Dravet syndrome is affecting the brain that yields similar Parkinsonian gaits?
So SCN1A is very present in the basal ganglia. We were actually part of our initial study, we were thinking of looking at performing PET scans to further look into this. However, with the COVID pandemic and everything, timelines have changed, but it's pointing in that direction a little. But definitely more studies need to be done. But cool. With their intellectual disability, and it's already very hard to get them to sit in a MRI scanner without all the complications that come with it.
Cool, thank you.
And a final question from Francis.
Hi, nice talk. I just wanted to ask, it's naive too, is based on the videos you showed us of the gait and so the measures, there's a lot of variability. And so I was wondering if there's sort of more sophisticated metrics you guys could use, where you put, say, sensors in different locations and, when you see the person walk, I mean, something, in rodents, for example, when they do behavioral studies, they just sort of monitor the movement. So given the huge amount of variability, is there a possibility to use more sophisticated metrics?
I think so. I think definitely for the patients that are more cooperative. Again, there's a cooperative issue, yeah. That's on here. So, and with that one patient I showed you with the dystonia, she's already leaning one way. So I think, it goes on a case-by-case basis there. But,
right. Yeah. I mean, that's exactly right. I mean, I don't know if there are sensors, if people would be willing to do that way they could kind of almost do what they want and then just sort of have those measures. But yeah, okay, appreciate it. Thank you very much.
Well, thank you.
Great, fantastic work. Thanks so much, Aranan. And we look forward to seeing what happens in the next wave of your data collection. Okay, great, thanks. So now we're going to move on to Brian Wu, and Brian is coming to us from Mohit Kapoor's lab. Brian.
Hello, everybody. My name is Brian. I worked with Dr. Kapoor over the last 4 years, and today I will be talking to you. Seems frozen. There we go. Talking to you about FP4 signaling and how it participates in pulmonary fibrosis. So just because I know a lot of people at Krembil aren't very familiar with fibrosis, I'll give a preface. Fibrosis is essentially the scarification process that translates healthy tissue into non-healthy, non-functional tissue that is primarily extracellular matrix. And fibrosis occurs across a multitude of organs and participates as a pathology in a multitude of diseases. That being said, you might ask the question why I'm focusing on the lungs. And the answer to that is multifactorial. First of all, in the lungs, fibrosis can act as a primary pathology. And second of all, you can imagine that if you are losing healthy lung tissue, you are losing respiratory function. And therefore, it has a very high patient mortality. And so there's a great need to investigate lung fibrosis in the field. Thankfully, as of 2014, there were 2 landmark studies that led to the release of 2 commercially available drugs for pulmonary fibrosis, particularly idiopathic pulmonary fibrosis, which is one subset of a large group of diseases. And these led to pirfenidone and nintedanib, which are capable of slowing down disease progression up to 50% in patients, which is a vast improvement to what we had before. But of course, a 50% slowdown is not the same as a cure and is not the same as a reversal of damage. So there is still room to grow in this field. Now, if we're looking at lung fibrosis, we have to look at the cell players. And one of the key players is the fibroblast, as the namesake suggests. And fibroblasts are primarily responsible for turning into the myofibroblast cell state and secreting the extracellular matrix that makes up the scar tissue. And this, it sounds simple at a surface level, but it's highly complex because fibroblasts interact with the microenvironment in very complex manners. They interact with other cell players in very complex manners. And myofibroblasts don't only consist of fibroblasts, which means figuring everything out is rather difficult. And to further complicate that, we can go down to the signaling level. And our lab was very ambitious and decided to tackle Ephrin and F signaling. Now, this protein interaction is very difficult to elucidate, especially in vivo, for 2 reasons primarily. Firstly, ephrins and Fs interact bidirectionally, meaning that the ligand ephrin interacting with the receptor F leads to distinct signaling cascades downstream both the receptor as well as the ligand. And so you can imagine in vivo, it's very difficult to parse out this interaction. Secondly, ephrins and Fs are highly promiscuous, meaning any ephrin or F receptor can interact with a multitude of partners. And so you can imagine determining a phenotype and pinning it down onto a single interaction is also incredibly difficult. Thankfully, we addressed a lot of these issues in our previous study looking at Ephrin B2, where we found that fibroblast-specific deletion of Ephrin B2 led to a protective effect against the development of lung fibrosis. And even more interestingly, we found that the cleavage of the Ephrin B2 ligand into the cell space, into the tissue space, was actually sufficient and required for the effect of pulmonary fibrosis activity. This suggests, of course, that the effect we're observing, the disease effect, is specific to the receptor, downstream of the receptor, because you can imagine if you're cleaving the ligand, then it cannot act downstream of the ligand anymore. That study opened up a few questions for us, primarily centering around the fibroblast, because while we identified the ligand, we have yet to identify the exact receptor. We were able to narrow it down to either FB3 or FB4, and for this study, we decided to go forward with FB4 because in the literature, FB4 is the primary signaling partner of Ephrin B2. So that was just the most likely candidate. We wanted to ask whether FB4 contributes to pulmonary fibrosis, and we wanted to ask whether there was a mechanism that we could discern. So to achieve this, we first had to create a mouse line that was a fibroblast-specific knockout for FB4 that was inducible by tamoxifen. And so we confirmed this by treating our mice with tamoxifen and observing the fibroblast expression of FB4, and indeed it is knocked out, as you can see here. We took our genetic mouse line and we hit it with a fibrosis model, a 2-hit model, The first hit removes FB4 in the fibroblast. The second hit induces fibrosis through the injection of bleomycin directly into the lungs. And then we have an endpoint where we can collect the lungs and subject the lungs to histology, where we can stain the total collagen of the lung blue. And collagen in this case is an indicator of disease activity because it's an extracellular matrix molecule. And as you can see in the wild type lungs, there's much more blue staining compared to the FB4 knockout lungs. And this is reflected in our scoring, which is the Ashcroft score, marking the severity of disease in affected areas of the lung. Now, Dr. Kapoor will be the first to attest that I am a rather fickle researcher. I am never satisfied with a semi-quantitative scoring system. And so I approached this from a slightly more computational aspect, where I took the ImageJ color deconvolution and I quantified the total collagen in the entire lung, the total blue staining. And from that, we were able to discern that there was actually a decrease in affected area of the lung, as well as a decrease in total collagen content in the entire lung. So this model seems to provide protection against pulmonary fibrosis in affected areas by decreasing disease severity, by decreasing area of effect, and also by decreasing total collagen content. Great, we have a phenotype, and now we need to investigate the mechanism. And since the mechanism is very unknown, we wanted to go with the most unbiased modality that we could think of, and that was next generation sequencing. So we took our mouse model that we had described previously, we isolated the fibroblast, subjected that RNA to next generation sequencing, and we managed to pull out 318 differentially regulated genes comparing the wild type mice to the knockout mice under fibrotic conditions. Now, that's a very long gene list. So with the thanks of Dr. Igor Juristika's lab, we were able to compare our differentially regulated genes back to a pathway repository. And we found that 7 pathways were specifically linked to the upregulated genes and 13 to the downregulated genes. We translated this into a visual pathway interaction map. And unfortunately, the upregulated gene list was rather uninteresting because many of the pathways there were incredibly generic, such as just cell metabolism. But thankfully, the downregulated gene list gave us a very, I would say, a gift of sorts, because we managed to pull out this semi-island of information that is all related specifically to extracellular matrix secretion and remodeling. And if we zoom in on that, we can see that there are actually 11 genes that link FB4 knockout in fibroblasts to extracellular matrix, which may be a reason why we have this incredibly fibrotic lung on the left and this well-protected lung on the right. Now, the first thing we wanted to do is validate our NGS findings because NGS does have a few caveats in it that we can address through another model. We went with an in vitro model where we treated fibroblasts isolated from mouse lungs with a pro-fibrotic cytokine, TGF-beta, and a pharmacological inhibitor of FB4. And we were able to validate 6 out of 11 gene candidates very strongly using this model. 2 candidates were not significantly different, but you can appreciate that there does appear to be a downregulated trend. And then 3 genes that had very poor response. We are currently working towards a functional validation using these findings to further solidify the link between FB4 and these identified gene candidates. And those experiments are currently underway. The other arm of interest that we really wanted to approach was addressing the translational potential of FB4 inhibition. And since we had a pharmacological inhibitor at our disposal, we wanted to see whether it had therapeutic potential. So we took our in vivo mouse model with a wild type mouse, we induced fibrosis with bleomycin, and we waited for disease activity to begin before we started our first treatment of our pharmacological inhibitor at day 5. And even though we treated these mice at day 5 with our inhibitor, we found that there was substantial protection of fibrotic activity using the pharmacological inhibitor, if you can look at the vehicle lungs versus the inhibitor lungs. And that was a very thankful result that we received, which we also looked at using our 3 modalities. And so in summary, we can say that fibroblast FB4 knockout does substantially protect against lung fibrosis, and it appears the mechanism is through extracellular matrix modulation, or genes that modulate the extracellular matrix. And so thank you very much for listening to my presentation. I am happy to answer any questions you may have. And thank you to everyone in the lab that has helped me along this journey.
Wonderful. Thank you very much, Brian. I'm not sure if you can turn on your camera for questions, if that's possible. Before we get to Liang's question, Francis has a clarification question, so maybe that would be helpful to start with.
Absolutely. Unfortunately, I don't have a webcam.
Oh,
I'm using
okay.
desktop.
Well, we'll just imagine how you look.
Yeah, no, you're a very clear speaker, so that's fine. You had shown, I didn't see what the slide number was, but when you had sort of the upregulated and downregulated genes and intersection, it was a little bit too fast for me to grasp, but I thought they were at the intersection, there were 25, but there seemed to be less in the separate. I don't know if you could go back to that slide and maybe just sort of kind of go a little bit more slowly there for me. But,
Certainly.
Yeah.
I'm thinking you're talking about
Yes.
this slide, correct?
Yeah. Yeah.
So you're talking about this one.
Exactly.
Ah, yes. So I didn't speak to this, but we also performed analysis where we took the entire gene list, not up and down regulated, and we intersected pathways to everything. And so these 25 pathways interact with both as well as down regulated. We ultimately decided to go forward with just the downregulated gene list because a lot of, it wasn't super clear, but a lot of these pathways were very generic pathways that were not super indicative of the mechanism.
Okay. Okay. Yeah. It was just confusing to me that just from a Venn diagram point of view, the intersection had more than the separate 30. Yeah. Oh, okay. All right. Okay. I see what you're saying. Okay. Got it.
Yeah.
Okay. Thank you.
Okay,
No
great.
problem.
So how about Liang?
Should I read the question out loud?
Sure.
Liang asked, what are proposed action mechanisms of drugs currently used for treating fibrosis? So that's very interesting. Both nintedanib and pirfenidone are pan-growth factor inhibitors. And so they target mesenchymal layer of the lung. They, so previous paradigm of lung fibrosis was very inflammatory approach, but these landmark studies really shifted the paradigm towards epithelial cell research as well as mesenchymal, and they're just pan-growth factor inhibitors.
Great, thank you. And Michael, you have a question?
Hello, thank you for your talk. I have a question related to FBs and the receptors in the lung. I guess there are probably other FB receptors in the lung, and I think you mentioned FB3. In the visual system, FNB2, FB3 has a strong interaction, I would say. So do you think there might be some sort of compensation mechanism? And if you would knock out FB3 together with FB4, you would even have a stronger phenotype, for the best of those lungs.
Certainly that is definitely has been on our mind a lot in this study because FB3 is definitely a very strong contender. And it was difficult going forward with just FB4. I think personally, since the FB4 genetic knockout as well as the FB4 inhibitor did have pronounced effects, there may be a compensatory mechanism, but there is still a protective effect showing. I believe a double knockout is something that is really interesting to go forward with. We were hoping to generate a double knockout, but we're trying to get our hands on the Floxed FB3 currently. It's definitely a question that's on our minds a lot, and unfortunately, I can't give you a solid answer because we haven't done it yet.
Of course, the drugs you're using might not even be specific only to FB4. They might target FB signaling, generally speaking, and you may, in this case, also target FB3.
Yes, certainly. This drug is marketed as an FB4 inhibitor, but I am certain it also inhibits other FBs at certain concentrations. So it would require some further validation that it's purely FB4 inhibitor.
Thank you.
Thank you. And I'm not sure if it's a very fast answer to Michael's question. We can see.
What are your thoughts on prophylactic inhibition? I've actually done prophylactic inhibition on both the genetic, on the inhibitor, sorry. And we actually, interestingly, found that prophylactic inhibition has a weaker protective effect than the therapeutic. So to me, that suggests that FB4 is not super active during the acute inflammatory phase of the of the lung fibrosis model, it participates more in the healing phase, so to speak, or the post-inflammatory phase.
Okay, thank you very much, Brian. That was very, generated a lot of really good discussion. So thanks very much for your talk.
Thank you very much.
I would like to thank all of our amazing speakers for a really wonderful afternoon so far. We're going to have a little bit of a break now, and then everybody is going to go in to see the even-numbered poster session. So to remind you to do that, go back to Gathertown for even-numbered posters. Then we're going to reconvene back here at 2:40. I just phenomenal talks all. And can I remind our oral judges to return their scores during this time? Because after all that, we'll have, after the posters, we'll also have another video and our awards and closing remarks. So see you all back here at 2:40 and enjoy the afternoon posters. Okay, so we are running a little bit behind, but we're just getting ready now to start with our second video put together by our wonderful team. And in this case, the last one was from the mentors experience, and this one will tell us something from our trainees' perspective. I hope they're not too tough on us.
Hello, my name is Alexandra Hadzikalimniu, a computational neuroscientist at Krembil.
My name is Anna, and I'm a PhD student in Dr. Krembil Tertalia's lab at Krembil.
I'm Lisbeth Ayub, and I'm a PhD student with Dr. Masi Moyadi at the Faculty of Dentistry and Dr. Mirfat McAndrews at the Krembil Brain Institute.
Hello everyone, I'm Mandana, and I'm a Master of Science trainee in the lab of Dr. Fellings at Krembil Research Institute.
My name is Wilford. I am a PhD student from Dr. River Lab at the Krembil Brain Institute.
Hi, my name is Stephanie, and I'm a PhD student in Dr. Robert Chen's research lab.
Hi, my name is Nadeem, and I'm a senior PhD student in Dr. Kroon Singh's lab, which is part of the Donald K. Johnson Eye Institute at KDT.
My name is John, and I work in Dr. Chandran's lab at the Schroeder Arthritis Institute. I study psoriatic arthritis because I want to help improve the assessment of psoriatic disease activity.
I'm working on a drug discovery for protein misfolding and a neuroinflammation involved in Alzheimer's disease.
I use neuroimaging to see how the brain processes pain, and particularly the hippocampus, a brain region involved in memory.
My research focuses on understanding how the brain differs in healthy older adults and people with Parkinson's disease when looking at balance and gait disorders.
I study the genetic cause of neurodevelopmental disorders, such as autism spectrum disorder, and more specifically, I try to find how the different genes associated with ASD interact with each other at a protein level.
My studies are in degenerative cerebral myelopathy, and I hope that one day I can improve the quality of life of elderly people.
What motivates me to go to work every day is going to Krembil and being surrounded by top researchers and clinician scientists, and this pushes me to be the best I can be every day.
Even the smallest result could potentially help someone, whether it's a scientist or hopefully a patient with one of the disorders that we are studying.
I've missed coming to work over the past year. These walls, they are literally filled with inspiration. Everyone around me is so focused and driven and alert and curious. And this vibe is contagious.
Research Day is important to me because it's a great way to learn about all the amazing research being conducted at Krembil.
It showcases the great research achievements made by our institute.
We're able to communicate our findings and understand the true implications and impact of our work.
I am very fortunate to have this opportunity and see the difference my research makes in patients' diagnosis and prognosis.
My goal is to develop models that can be used in industrial and clinical applications and improve the lives of many.
I enjoy my time at Krembil because we have built a community, and I get to interact with patients and analyze brain scans and sometimes do fashion shows. My ultimate goal is to become a neuroscientist.
Hope you enjoy Research Day.
I hope you enjoy Research Day.
Thank you, thank you, thank you so much. That wasn't too rough at all, actually. I think we all, the PIs escaped without scars. It was wonderful hearing from your perspectives, and especially the theme that I heard a lot of times was about community. It's amazing that we're able to kind of keep the community together, even despite the pandemic challenges. So I believe, unless Carly tells me otherwise, nope, she has just texted me, that I get, before introducing our fearless leader for some final words, I get to actually announce the awards for presentations today. The office will be in contact with you regarding what the award is. It's a job. No, I'm only kidding, but that would be nice. Okay. So for postdoctoral fellow, the award goes to Scott Rich. For our graduate student awards, we have 3 awards. In first place, Brian Wu. In second place, Nadim Murtaza. And in third place, Akshay Gurdita. So congratulations to our awardees. Congratulations to all of our wonderful presenters, posters, as well as oral presentations. I certainly enjoyed the opportunity to have more interaction in the poster sessions, although I will say they weren't long enough because we definitely needed more time for all of the great work that was being described. So once again, the office will contact you guys for the awards. Congratulations to everyone. I had a wonderful day, and now we will finish it off with hearing some words of wisdom from our illustrious leader, Dr. Donald Weaver.
Thank you. Congratulations to all of our winners. And as I like to say, if you work at Krembil, you're already a winner. I certainly hope that you've enjoyed today's event and that you've had a chance to wander and to mingle in the different rooms, meeting up with some of your colleagues, as well as perhaps interacting with some new ones. You know, and in many ways, it really is impossible to replace the experience of gathering in person. But with this new platform, GatherTown, our hope was to get as close to this as possible. And thanks to the wonders of modern technology, it almost felt like we were all back together today, almost. And there certainly is comfort in coming together. And if there's one thing that I have seen and felt over the course of this pandemic, it's the power and unifying force of the Krembil community. I know that I have said this many times, and you'll hear it from me over and over again, but I want to thank each and every one of you. Over the course of the past year and a half, I really have been truly amazed and so very proud of the resilience and dedication that you've all shown in these tough and unprecedented pandemic times. And if there's 2 take-home messages that really have come clear to me over the course of the past 15 months, it's these. Number 1, no scientist achieves anything truly alone. We need the right people around us with the right experience. And more importantly, we need people with the motivation and the drive, not only to ask the right questions, but to find the answers. Science, successful science, it's a team sport. And let's not forget that. The second take-home message is that science, it's the future of our society. Indeed, it's the future of humankind. And, you know, good science is going to be important as we go forward. So we should appreciate that there's a momentum going to be happening from the wake of the COVID pandemic. And if there's anything positive that's going to come out of COVID, it's going to be that we're shining a spotlight on science, and that hopefully this will have a beneficial effect on future biomedical research. I don't think we should ever lose sight on just how important science is in society. And I think that we should, you know, appreciate that we all have to work together to find ways to build on the momentum that has arisen from COVID and to push discoveries further, not just for COVID, but for all the very important disorders and diseases of brain, of joints, of eyes that we study at Krembil. Now, you can help in doing this by taking advantage of the science communication training opportunities through our Krembil Public Affairs and Administrative teams, learn how to communicate more effectively with the public, media and donors, and better promote your work. We really must continue to break down silos between science and the public, and to better inform those in our communities, and to drive future support and advocacy for our work for good science. Because of COVID, we now know the importance of strong and clear communication and what happens when you don't have it during a public health crisis. And we have seen lots of examples of both. Please remember the good ones. Finally, a big thank you to our Training Affairs Committee, the Krembil Administration and Public Affairs for putting together a truly excellent event today. In particular, I'd like to thank Nadeem Murtaza and Samir Patel for building our virtual event space in Gathertown, and for the training sessions that they ran for the speakers. It takes a village to organize Research Day each year, and a lot of details go into the planning and executing of this, especially if we're doing it in this virtual setting. Thank you also to our dedicated team of staff and volunteers, and to all of you for coming out to celebrate the research accomplishments of our amazing Krembil community. We're all in this together, and we're certainly not through this. COVID's not done with us yet, and we have a ways to go. But please, let's remember the importance of science, the importance of the Krembil community, and everyone, please remember to stay safe. So once again, thank you ever so much. I really appreciate your involvement, and I hope that you have found today rewarding and enriching. Thank you.